I'd like to welcome Ash Patel, who's a vascular surgeon at Guy's and St Thomas's, to the stage. Ash has worked in the BRC or with the BRC for many years and is currently leading on some of our trials in cell and gene therapies. And he's going to share some of his first-hand experiences of manufacturing these novel therapies. So Ash, welcome, and over to you.
Okay, thank you very much for the invitation, and good morning, everyone. So I'm an academic vascular surgeon at King's College London and Guy's and St Thomas's. Next slide, please. And I'd like to take you through a bit of a whirlwind tour through what's been happening in the last year in the GMP unit. I've been working with BJ Modrai for many years on cell therapies for peripheral arterial disease. And I'll talk about what we've been doing in that field and how we recently repurposed our work for COVID-19. So next slide, please. So we have been working for a while in this area and an interest in delivering cells to regenerate new blood vessels in patients with ischemic legs. And it's quite a complex process, and we have been watching other clinical trials in this area, and we've seen quite poor results, mainly because the process of growing new blood vessels in ischemic tissue is quite complex. Next slide, please. And we have a special interest in monocytes and macrophages, and we showed recently that these cells are pro-arterial and angiogenic. There's a certain population of these cells which, when we deliver them into ischemic hind limb models in mice, the limb salvage rate is quite impressive. And we were able to translate this into a cell tracking study within the BRC a few years ago, where we isolated and delivered these monocytes into patients with ischemic limbs. And we showed that the retention was quite poor and that these cells are washed out within 3 days. So we had to come up with a new way of manufacturing high numbers of these cells so that we could essentially inject these cells as repeated doses. So next slide, please. And so we use the notion that mesenchymal stem cells are able to immunomodulate other cells. And when we co-culture mesenchymal stem cells with patient monocytes, we can upregulate markers which give them this angio and arteriogenic property. And again, in our in vivo models, we showed that these what we call MSC primed monocytes can revascularize ischemic tissue. And what they do is they induce arteriogenesis, which is important for blood vessel, for taking blood down into ischemic tissue. So next slide, please. So in the first wave of the pandemic, as we were about to translate our work for peripheral arterial disease into the GMP unit, we noticed that patients who were being discharged with COVID-19 were coming back and being re-referred with pulmonary fibrosis or interstitial lung disease. And this was going to be an endemic because of the high numbers of patients who are developing lung disease. So next slide, please. And we decided to go back and look at our data, and we saw that in our ischemic limbs in our mice that the fibrosis levels were markedly reduced when we had injected these MSC primed monocytes. So we carried out some more experiments to confirm that these cells do actually reduce fibrosis, and they also protect small airway epithelial cells when they are damaged with cisplatin. So next slide, please. And as a result, we decided to repurpose our cells in 2020 and instigate the Monaco cell therapy study, so monocytes as an antifibrotic therapy in patients who recovered from COVID-19 as a phase 1 safety study. And there were secondary objectives to look at efficacy in terms of lung function and quality of life. Next slide, please. So I'd like to talk to you about manufacturing and what it takes to go from the bench to the bedside. And the key part of this is the Investigational Medicinal Product Dossier, the IMPD. Without this, the MHRA will not approve your product. So next slide, please. So there were two main parts of this. One was to generate a working bank or mesenchymal stem cells, and the other one was to then take these monocytes and engineer them and show that we could reproducibly turn these into antifibrotic cells. And so next slide, please. And we were fortunate to receive funding from the Rose Trees Trust, and then also the BRC and Guy's and St Thomas's back in May of last year with a COVID-19 contingency award. So the first key step was to generate these mesenchymal stem cells. So next slide, please. And we worked with a company in the States called Rooster Bio, and the important part here was that they had a validated process where we essentially needed to replicate that in our GMP unit. So we went from very small T75 type flasks into large scale stacks. We trained the team, the fantastic team within the GMP unit to use these stacks, which they hadn't used before, and to essentially bank large numbers of mesenchymal stem cells that were ready for use in these patients. Next slide, please. So having generated the mesenchymal stem cells, we then had to do our 3 engineering runs to confirm that we could generate these monocytes. So next slide, please. And this process was very different. We did not have a validated process for blood, and there wasn't a sort of oven-ready, off-the-shelf method to isolate monocytes from whole blood, and that was our plan. In the lab, we had done this with using Ficol, and we knew that we could do this using leukapheresis product, but the companies said that there was no option to do this with blood. So we had to carry out some scale up and some processing work within the GMP unit to take what essentially was the lab process, using novel equipment such as a Lovo and the Clinimax machine to make sure we could isolate these monocytes in blood. So next slide, please. And so having carried out 3 engineering runs successfully in August of last year, we had to then go and develop potency assays before we submitted our IMPD. And we worked closely with our QP to ensure that this was all done in good time. So next slide, please. And these potency assays are very important. It gives the MHRA some clarity on the fact that these cells actually have function in these patients, and we had to ensure that when the cells are frozen and then thawed, that the surface markers are retained and they still function. And again, we had to work with industry and use benchtop devices to ensure that we could carry out these assays reproducibly and reliably. So next slide, please. And following all that work, towards the end of last year, we were able to compile our IMPD. And I think the key message here is that there are so many documents and there is so much work that happens behind the scenes in a GMP lab, and the manufacturing team do so much work that you don't really see as you're trying to generate your product, that having a team working on this is essential. Next slide, please. And so towards the end of the year, around December, we were able to put all these data together and submit them to the MHRA for approval. Next slide, please. So just to summarize, so what's happened since? In December, we had MHRA approval to start the trial. We had to pause because of the second wave of COVID, and we started recruiting. We had green light in March of this year. The first patient was dosed on the 7th of April, and we've successfully dosed all the patients. The last patient was dosed just this month. And the plan now is to follow these patients up over the course of a year. They'll have lung function tests and CT scans, and hopefully we will see whether or not these cells have any effect in reducing their fibrosis. These are otherwise healthy patients who, before they developed COVID, were fit and healthy and had no respiratory problems, and they really are now disabled by their breathing. The future plans are to then perform a dose escalation and safety trial to determine what the maximum tolerated dose is in this patient group, and we have plans for this hopefully in the near future. So next slide, please. In terms of manufacturing, one of the key challenges is to ensure that you can produce a product reliably every single time, and we're working on automating our process to minimize the risk of batch failure. And I think in the future that's an important consideration for cell therapies, to make sure that we can create reproducible batches. Next slide, please. So all this work could not have been done without a huge team. That includes people in our laboratory, in academic department of surgery, but also within the GMP unit, the manufacturing team, the production scientists, and the quality team as well. And then also the clinical research facility were instrumental in making sure as soon as we had a viable product and MHRA approval, we could then start recruiting patients very quickly and dosing them safely. So I'd like to thank everyone who was involved in this trial, and hopefully we can give you some updates in the near future.